and welcome to my presentation where I'm going to talk about my PhD project where I'm trying to characterize the tumor microenvironment associated to a new pancreatic cancer tumor subtype that we found by analyzing translation, mRNA translation. For this, we make extensive use of independent component analysis, or ICA, which is a very good way of separating mixed data into its constitutive source signals. For this, ICA, like principal component analysis, is a matrix factorization method, where mixed data is split into gene contributions, representing the gene relevance for each component, and sample component scores, which are just indicating the component abundances across samples. Pancreatic ductal adenocarcinoma, or PDAC, is a lethal disease, characterized by having a very abundant stroma and generalized immunosuppression, where fibroblasts highlight by their abundance. All of the efforts trying to characterize PDAC at the level of transcription converge in the detection of classical and basal-like subtype, which are prognostic but do not offer any therapeutical advantage. On the other hand, the studies focusing on the study of protein synthesis, the process by which mRNA gets translated into proteins by the ribosome, revealed that PDAC has a very altered mRNA translation, as we reviewed in this recent article. And this motivated us to explore for the first time the classification of PDAC at the mRNA translation level. So for this, we evaluated the translation efficacy, calculated using gene-wise linear models, comparing the total mRNA obtained by normal RNA sequencing with the translated mRNA, which is obtained by isolating and sequencing those RNAs associated to ribosomes and thus undergoing translation. For this, we have to apply a very complex preprocessing that I will not cover here today. And to evaluate this in PDAC, we use patient-derived xenografts, or PDX models, a hybrid model system where human tumors are grafted inside mice, which later, using tools like ISMB, allows the computational separation of the reads that belong to the human tumor from those that are from the mice stroma. By focusing in the human tumor translation efficacy and applying ICA on it, we found ISRACT, a new tumor classification not associated to basal classical, and in turn correlated with the translation efficacy of the stress factor ATF4, as well as to a global downregulation of translation. Model after in vitro characterization revealed that actually ISRACT tumor cells have a persistent activation of the integrated stress response, as well as an increased chemoresistance, making this classification of therapeutic relevance. For this, in my PhD, what I've been trying to do is to characterize this stroma using transcriptomics and also looking at the translation efficacy. In both cases, applying ICA and looking first at the sample component scores to see if there's a component related to ISRACT, and then trying to characterize, looking at the gene contributions, trying to characterize what the component is actually representing. When looking at the stroma transcriptomics, we found IC4 as the component that captured the traits of ISRACT, also having a high correlation with the presence of estimated lymphocytes. Enrichment analysis of the gene contributions further support the involvement of immunity, and by selecting the 5% top contributing IC4 genes and looking for described protein-protein interactions, focusing on highly connected regions or models, shows that IC4 is capturing response to interferon stimulation as well as antigen presentation, supporting immune recruitment. When looking at the stroma translation efficacy, IC6 lands as the best component representing the tumor phenotype. And when analyzing the correlation of the components with the transcription levels of RNA binding proteins, or RBPs, the proteins that control the fate of mRNAs including translation, IC6 seems to be the only component that is specifically correlated with the level of these compared to randomly selected genes. In particular, positively with M6A mRNA modifications and mRNA degradation, and negatively with mRNA processing and splicing. When we looked at the 5% most down-translated genes of IC6 in the gene contributions and their first network in a context of protein-protein interaction network, we found a model related to fibroblasts and matrix secretion, where lumican is the only down-translated gene, and it has been described to affect the tumor microenvironment in many ways, including promoting inflammation, suggesting that perhaps a downregulation of lumican could be promoting this characteristic immunosuppression. To try to see what could be regulating Lumican, we look at experimental CLIP databases, and we found out that TARBP was the most likely regulating RNA binding protein, with conserved targets in Lumican and described to bind preferentially to M6A methylated mRNAs, as well as drive mRNA degradation to CCR4 node, connecting with the results we saw before. Thus, in a nutshell, we believe that secreted factors from the tumor are recruiting immune cells, which in turn are secreting interferon gamma, affecting the local tumor microenvironment. This would drive, for instance, the polarization of CAFs to antigen-presenting CAFs and antigen presentation. In turn, IC6, captured by translation efficacy, could be capturing the downstream immunosuppression that is being described to be driven upon fibroblastic antigen presentation, where perhaps Lumican downregulation could play a role. Yet, there's still many things more to explore, starting with finding this classification in the human cohorts and in single-cell RNA-seq, which we recently did, trying to validate the results of immunity as well as find candidates for in vitro validation. As well, we would like to further explore translation by looking at RBP target mRNA networks and translation-specific tools like Anota2seq tools. Last but not least, I want to thank you for your attention, the members of my two teams at the CRC team Toulouse, as well as the collaborators and sponsors. Thank you very much.